Hi, welcome to Bioreactors channel. Now I am going to take class from 12th standard Botany. Now let us discuss about chapter 4, topic fermentation. What is fermentation? Fermentation is the metabolic process. In this process, organic molecules are converted into acids, gases. See here, organic molecules converted into acids or gases. This process is called fermentation. Okay. This conversion process need microorganism. This conversion process need microorganism. Microorganisms, it may be bacteria, yeast or fungi. Organic molecules converted into acids or gases in the presence of microorganism, but in the absence of oxygen. In the absence of oxygen. This process is called fermentation. Okay. What is organic molecule? Do you know that? Molecules that are produced by living system, that is called organic molecules. Okay, molecules produced by living system, that is called organic molecules. Okay, lipids, proteins, carbohydrates, these are all the organic molecules. Okay, for example, sugarcane. Sugarcane plants coming under living system. Okay, sugarcane contains starch. Sugarcane contains starch. Starch is one of the organic molecules. Okay, starch is one of the organic molecules. Louis Foster demonstrated fermentation caused by yeast. Louis Foster demonstrated fermentation caused by yeast. But fermentation not only caused by yeast. Fermentation can also caused by bacteria, fungi. Okay. What is zymology? Do you know that? What is zymology? Study of practical uses of fermentation. That is called zymology. Study of practical uses. Study of practical uses of fermentation. That is called zymology. Understand Is this clear? Okay. Fermentation has some practical uses, and fermentation helps in food industries, beverage industries. In food industries, we can make soft bread by using fermentation process. In beverage industries, we can produce soft drinks and alcohol. Alcohol produced by using fermentation process. Understand? Is this clear? I already explained you. In fermentation process, organic molecules converted into acids or gases. This acids or gases helps to preserve the vegetables and dairy products. Okay, these acids and gases helps to preserve the vegetables and dairy products. Understand this clear? Okay, let us move on to next one, fermenter. Next we are going to discuss about fermenter. Next we are going to discuss about bioreactor. What is bioreactor? Is also called fermenter. Fermentation process takes place in fermenter. Okay, fermentation process takes place in fermenter. What is fermentation? Substrate converted into product by using microorganism. Okay, this process is called fermentation. What is substrate? What are the things we are going to add? That is called substrate or raw material. Raw material is the substrate. Finally, we get product by using microorganism. This microorganism convert the raw material into our desired product. Understand? Is this clear? This process need optimum environment. Okay? Fermenter should give optimum environment. If we want to live in this world, we need optimum environment. Extreme temperature, we cannot live. And very low temperature, we cannot live. So, we need optimum environment to live here. Like that, microorganism also need optimum environment. This bioreactor should provide optimum environment. This bioreactor should maintain the aeration, agitation, temperature, pH, aeration, need of air, agitation, mixing process, and it should provide optimum temperature and pH for growth of microorganism, because microorganism only convert the substrate into product. This fermentation process does not occur without the microorganism. Understand? Is this clear? Fermentation process does not occur without the microorganism. I will say one example for you. I will give one simple example. Milk converted into curd by using this fermentation process. Understand? Is it clear? See here. Milk, example, milk converted into curd by using microorganism. This process is called fermentation process. Milk is the substrate here. Curd is our desired product. In this process need microorganism. One more example. Grape juice. Grape juice converted into alcohol. This process need microorganism. This microorganism convert the grape juice into alcohol. Grapes are the substrate here. Alcohol is the required product. This process is called fermentation process. This fermentation process takes place in fermenter or bioreactor. Understand? Is this clear? Substrate converted into product by using microorganism. This process is called fermentation. This fermentation process takes place in fermenter. Fermenter should provide optimum environment for the growth of microorganism. Process contain two processes. One is upstream process, another one is downstream process. First one is upstream process, another one is downstream process. What is upstream process? What are the process we are going to do before start the fermenter? Before start the fermenter. This process is called upstream process. Understand, is this clear? Before start the fermenter, what are the process we are going to do? That are all coming under upstream process. Okay, what is downstream process? After fermentation. After fermentation, what are the process we are going to do? That are all coming under downstream process. Understand, is this clear? Okay, let us discuss about upstream process. In upstream process, we have to sterilize the fermenter. First step is we have to sterilize the fermenter. What is sterilization? Do you know what is sterilization? Kill the microorganism. Kill or destroy the microorganism. That is called sterilization. We have to kill the microorganism in the fermenter because it will cause any other reaction. So we should kill the microorganism in the fermenter. Next step is we have to sterilize the culture media. We have to sterilize the culture media. What is culture media? Culture media provide necessary things needed for the growth of microorganism. Okay, for example, nutrients, water. These are all very important things for the growth of microorganism. Culture media provide the necessary things needed for the growth of microorganism. Next step is we have to add the microorganism into the culture media. We have to add the microorganism into the culture media. These all the steps coming under upstream process. Understood, is this clear? Next we will discuss about downstream process. Already I explained you. After fermentation, what are the process we are going to do? That are all coming under downstream process. Distillation, centrifugation, filtration, solvent extraction. Solvent, these are all coming under downstream process. See here, what is distillation? We have to purify the liquid by using distillation process. We have to purify the liquid. Purification. Purify the liquid by using distillation process. What is centrifugation? We have to separate the product based on the densities. We have to separate the product. Separate the product based on density. This is called centrifugation. Next step is filtration. What is filtration? This is also the separation process. We have to separate the product by using filter media. Filtration also used to separate the product by using filter media. Finally, we extract the product. Finally, we extract the required product. See here, substrate, converted into desired product by using microorganism. This process is called fermentation. This process is called fermentation. This fermentation process takes place in fermenter. Understand? Is this clear? Thank you. If you need any other topic, give your feedback in the comment box. We will see in the next video. Please subscribe my channel.